So the main question that is being addressed in figure 3 is whether or not vascular density and parasite coverage are observed to have any difference in germ-free versus pathogen-free mice. So we'll start up here with figure 3A. Figure 3A uses two photon imaging to compare the brain vasculature of both germ-free on this side and pathogen-free mice on that side, 20 to 80 micrometers below the dura mater. Figure 3B over here uses imaging to compare the brain vasculature 120 to 180 micrometers below the dura mater. Down here in figure 3, this uses quantitative data to compare the blood vessel density 20 to 80 micrometers below the dura mater. And then right here in figure 3D, that uses quantitative data to compare the blood vessel density 120 to 180 micrometers below the dura mater. And then if we go over to this side, figure 3E, we see that this uses immunofluorescence staining to represent the parasite coverage in the cerebral cortex of pathogen and germ-free mice. So basically this figure shows that there is no significant difference between the vasculature density in the brain of the germ-free adult mice and the pathogen-free adult mice. There is also no significant difference in the parasite coverage between the germ-free mice and the pathogen-free mice. So these results exclude the possibility that high brain-blood barrier permeability in germ-free adult mice was caused by higher vascular density or decreased parasite coverage.
Looking at the experimental design of this study, we can see that the researchers used two different groups of mice to study the impact of gut microbiota upon the permeability of the blood-brain barrier. They used a group of mice that were considered the germ-free mice that had never been in interaction with living organisms and living microbiology. And they used a group of pathogen-free mice that had never been introduced to common diseases for mice, but had been introduced to microbiological cultures. In this study, the independent variable was that of the group of mice that they were drawing from, and the dependent variable was that of the permeability of the blood-brain barrier and the parasite density. The researchers used this study in this figure 3 to demonstrate as a control that the blood-brain barrier permeability was not affected by the presence of gut microbiota. The control within this figure itself was the depth of the dura mater that was studied. Figure A and C represent the same level of in thickness that was used in the dura mater that was studied to view the permeability of the blood-brain barrier. And figure B and D represent the second layer of thickness. Now this thickness was verified by using second harmonic generation microscopy to ensure that it was the same level within the two different types of mice that's being studied, to make sure there was not different thickness in the blood-brain barrier.
So the scientists used intravital two-photon microscopy to rule out the chance that the high blood-brain barrier permeability in germ-free mice was caused by vascular density in the brain. Intravital means that the mice were not killed during this experiment. They were totally alive. The two-photon microscopy is a fluorescence imaging technique that allows imaging of live tissue up to about 1 millimeter in depth. And this two-photon microscopy was used in figure 3A and figure 3B. In figure 3, the scientists are looking at the blood vessels in the subdural mater, which is the potential space below the dura mater, which is the tough outermost layer of the brain. Figure 3E, which is right here, shows an immunofluorescence staining of the parasite coverage in the cerebral cortex of the germ and pathogen-free mice. CD13, which was used in figure 4E, is a transmembrane protein which was used to mark the parasite coverage, which is the endothelial lining of capillaries. Lamidin, also used in figure PGA4E is a protein present in the layer of the lining of a capillary, which was used to mark epithelial cells which form the lining of the blood vascular system.